Good day, everyone. We are Group 3 reporting about Cytomegalovirus. Etiology.
CMV were first noticed in 1881 by German scientists who thought they represented protozoa. After viruses were grown in cell cultures, the German scientists Wheeler, Smith, and Rowe, they independently isolated and grew CMV from man and mice in 1956 to 1957. Cytomegalovirus or CMV can only be passed on when it's active. The virus is active when you get CMV for the first time, like young children, when it is reactivated because of weak immune system, been reinfected with different strain of CMV.
So the common causes of acquiring CMV or cytomegalovirus is through close contact to someone who is infected by CMV, can be passed through sexual contact, if not protected, through semen, contact with other body fluids, including respiratory droplets, or we have saliva through coughing or sneezing, blood transfusion, breast milk, urine and stool, and other is transplanted organ. Other type is the congenital CMV, and this can be passed by a pregnant woman that has active CMV infection to their unborn baby.
Human cytomegalovirus, or HCMV, the prototypical member of the beta herpes viridae, the subfamily within the herpes viridae, and is found on about 90% of the population worldwide. HCMV has a characteristic three-layer architecture: an outer lipid bilayer envelope, an inner pseudo-icosahedral nucleocaspid, and a middle pleomorphic tegument compartment. The virion is usually spherical in composition, and the average size can range from 200 to 300 nanometers. In HEMV, as well as other herpes viruses, the nucleocaspid is formed by the complex interaction of virus caspid proteins. There are four major components of HEMV caspids: the major caspid protein, the triplex dimer, the triplex monomer, and the smallest caspid protein.
So this is the microscopic structures of the HCMV C1 capsid in C2 portal and CVSCS. So as what you have seen in picture A, it is the central slice of the capsid asymmetric reconstruction showing the unique portal vertex and the package dsDNA. And for the picture B, it is the zoom-in view of the box region in blue A, showing the interactions among portal protein, capsid proteins, and the pDNA. So in the top view, or in picture C, it is the portal vertex. And for the picture D, it is the structural organization of the portal main body, the 10 helix anchor, and the portal encircling, or pDNA. And for the E, it is the model of PUL-104 colored by residue from the N-thermos. That is the blue part. And to the C-thermos is the red. And for picture F, it is the unsharpened density map of 1 CVSC binding penton vertex. And lastly, for the G, it is the models of the 3 CVSC proteins of PUL-77 and PUL-48 and PUL-93. Each protein is rainbow colored by residue from the N-thermos blue and to the C-thermos red.
The portal protein is colored as in the contour levels of all protein density maps are indicated. The pipe and the plank depiction of the portal, the C6 turret, the C12 main body, and the C5 10 helix anchor are in magenta, cyan, and blue respectively. Atomic model of the portal turret and main body. Left top view of the portal turret showing the 6 coiled coils. Right side view of the portal turret and main body. Two conformers of the 12 PUL-104 molecules are highlighted in blue and pink respectively in sit density map of the portal vertex, and the model of the box region showing density connection between the turret main body as indicated by red arrows.
Pathophysiology.
Cytomegalovirus may induce a chronic inflammation leading to atherosclerosis and age-related diseases. In utero, infection is usually severe, causing neurological damage, high morbidity, and mortality in immunosuppressed individuals. The pathological hallmark of CMV infection is an enlarged cell with a viral inclusion bodies. Cells that exhibit cytomegaly are also seen in infections caused by other beta herpes virinae.
Cytomegalovirus infection is a common herpes virus infection with a wide range of symptoms, from no symptoms to fever and fatigue to severe symptoms involving the eyes, brain, or other internal organs. Once CMV is transmitted, the primary infection clears. The virus remains dormant in myeloid cells. Vital replication and reactivation are contained primarily by cytotoxic T-cell immunity. When reactivation occurs, virions are released into the bloodstream and other bodily fluids, leading to the presence of symptoms, predominantly in immunocompromised patients.
Clinical manifestations of cytomegalovirus. In babies, congenital CMV occurs wherein a pregnant mother that has an active cytomegalovirus passed on the virus to the unborn child. The manifestations are jaundice, yellowing of the skin or whites of the eyes, microcephaly, where a baby's head is smaller than expected when compared to babies of the same sex and age. Third one is enlarged spleen, purple skin or blotches, visual loss, and seizures. In adults, the common clinical manifestations are fever, muscle ache, skin rashes, swollen glands, tiredness, and sore throat.
Histologic examination. Cytomegalovirus is a common herpes virus that affects 50% of people by the age of 40 years. Acquired cytomegalovirus can spread between people through bodily fluids such as saliva, semen, blood, urine, vaginal fluids, and breast milk. A person can also contract the virus by touching a surface that has the virus on it and then touching the inside of the nose or mouth. Most humans contract the virus during childhood at daycare centers, nurseries, and other places where childhood come into close contact with each other. However, by this age, the child's immune system can normally deal with an infection. Recurring cytomegalovirus can occur in people with a weakened immune system due to HIV, organ transplantation, chemotherapy, or taking oral steroids for over 3 months. Congenital CMV normally occurs when a female contracts CMV for the first time either during pregnancy or shortly before conceiving. Occasionally, a dormant cytomegalovirus may occur during pregnancy, especially if the person has a weakened immune system.
Histologic examination of cytomegalovirus infection. Hematoxylin and leucine staining for CMV. CMV infected cells are indicated by yellow arrows, as you can see in the picture. Cells infected with CMV are larger with a thickened nuclear membrane and granular intracytoplasmic inclusions. Typical owl's eyes or basophilic intranuclear inclusion bodies are representative of active cytomegalovirus replicating nucleoprotein cores.
And that is all for our report. I hope everything is clear and understood. Again, we are Group 3 reporting about cytomegalovirus. Thank you.